Yeah, hello and welcome. In this special edition, I give an update about Corona. Yeah, quite a while ago, namely beginning of May, I talked about coronavirus in the context of the lecture Applied Microbiology and vaccine developments and so on. Now, this is light years away from the situation. Now we have vaccines approved, and therefore it's time for an update. And this update is in the context of the lecture Microbiology, the first semester. And I will talk about the virus and I will talk about vaccines, state of the art. Are they safe? How do they work? And this will be the content of this little video. So if we talk about viruses, the first thing to look at is the viral structure. And the good news is there is not much to say about the viral structure. So you see on this chart, we have viruses which are enveloped and which are naked. And if you look here, you see that the structural components of an envelope virus is that it has a lipid bilayer as an envelope, and in this lipid bilayer you have glycoproteins inserted. This lipid bilayer usually comes from the host membrane, so the cytoplasmic membrane. Then inside of this lipid bilayer we have capsid, and within that we have the nucleic acid inside. And this nucleic acid plus capsid is also called the nucleocapsid. So naked viruses just consist of this nucleocapsid, basically, and enveloped viruses have in addition an envelope and the glycoproteins inside. Viruses contain either DNA or RNA, not both. So for classification, obviously, you take this into account. So DNA viruses are viruses which have DNA inside, believe it or not, and RNA viruses are viruses which have RNA inside. And you see then you have a scheme, then you have enveloped DNA viruses, RNA viruses, naked viruses, DNA, RNA, and different viruses you have in this group. An example, RNA viruses enveloped is Sars Cov-2. An example for a naked DNA virus are adenoviruses, which are provoking the common cold. Okay, so that's about simply the structure. And as we have DNA, RNA, there's also a classification according to how the mRNA is being produced. And it's not DNA, RNA, it could be double-stranded DNA. This is the thing what you would say it's the normal case for DNA, but there are also viruses which have single-stranded DNA. So of course, then they have first to synthesize double-stranded DNA before they can start to transcribe into the mRNA. Now, RNA viruses are even more diverse. So you can have double-strand RNA viruses. RNA, as you know, is usually single-stranded. Then the single-stranded RNA, if it's a single-stranded RNA, it could be a positive strand. Therefore, you could transcribe directly from this. And a single-strand RNA, which is negative, means that from this strand you cannot transcribe. You need to translate it first into the positive strand. So then you have also single-stranded RNA viruses which replicate via a reverse transcription step. Then DNA is produced, this DNA is integrated, and this DNA then is translated to have this transcribed into have this mRNA. And you have also DNA viruses which have an additional reverse transcription step. So this is just for the sake of completion. The coronavirus, as we said, is a single-strand RNA plus virus. So let's talk about corona, and of course also let's talk about therapies, especially vaccines. So here we go. So this is the replication cycle of corona, and you see it's quite a busy slide. So this is your virus particle, and the first step is, of course, an attachment to the host cell, which happens via receptor binding to the ACE2 receptor. And then this virus particle is internalized, and the membrane fusion, this is interesting, doesn't happen at the cytoplasmic membrane. So the virus, of course, needs to release its especially genetic material. This happens on the level of an endosome. And that this can happen, there are a couple of proteases, host proteases involved, including this host protease called TMPRSS2. So this is required in order to achieve this fusion. If you block this molecule, we will see later on, you will block viral entry. The single-strand positive strand RNA is then released. And then you have the replication. And this replication is, of course, a viral RNA replicase. It's an RNA replication. This happens, as has been shown now, as kind of shielded complexes because RNA is not very stable. So this protects the RNA somehow. Then if you produce then your new mRNA, so to say, you can produce then the proteins you need. So by the way, this for the replication, it's produced at the beginning. It's a plus strand, as I forgot to say. So you can directly translate that into the replication complex. You see also you have, this is a large precursor protein, which is then Kleve, which is also typical for viruses, that you have often just one open reading frame. Remember, we are talking about eukaryotic viruses where you usually have no internal ribosomal entry sites, something what you have learned in cell biology or you will learn in molecular biology. Now, this is your genomic RNA. You have then different spliced variants, and these then produce the proteins, which happens inside of the cell. And this happens then, many of these proteins are directly synthesized in the endoplasmatic reticulum. And then you have a compartment, which is a kind of an interim compartment between endoplasmatic reticulum and Golgi. And there the assembly starts and the virus buds, so to say, inside of the Golgi system, and then it's secreted and released. So this is the replication cycle. And of course, the knowledge of this replication cycle is relevant if you want to develop a drug against this virus. And you already see that there are a couple of points added here where you could potentially block the virus. Once I already said, if you use an inhibitor to block this, these proteases which are required for membrane fusion, you will block entry. But also you have inhibitors of, for example, of the viral protease, because this is something which is required to cleave that protein, which is not which are of course special. And you have of course the viral RNA replicase, which doesn't exist at all in eukaryotic cells. So this is also a nice target. And for example, you could use nucleotide analogs, which are incorporated just by the viral reverse transcriptase, inducing then errors, and then the virus is no more can no more replicate. And this is an example is remdesivir. First, check whether there are questions. The single DNA and RNA is only TU base. One thing. The other thing is that it's ribose instead of desoxyribose. The difference between, it doesn't look so scary. How does this virus production harm the cell? Okay, so the pathogenicity of the virus, there are several things which makes this virus pathogenic. Usually the cells do not survive a viral infection for 48 hours. I might upload the paper where I've taken out this Nature review, where I've taken out this graph. Of course, it's very interesting to look at the pathogenic mechanisms, but you can imagine if you have enough virus RNA in the cell and the cell just produces viruses, it's an extreme stress for the cell, and the cell will not survive that at the end. So for the people this is a good question, we come to this question. So for the people that have never been infected, the neutralizing antibodies would be enough. Good question. Just remember it. We will answer this question in a second. Okay, so maybe just showing you one original data which has been published in April. They looked at this proteases at the first step, and you see at least the German students might recognize this name. On this paper, you have also Christian Drosten. So Christian Drosten is a virologist who doesn't just talk about corona, he also publishes a lot, and he's really an expert in this field. And as you're a scientist, you have to look on the data. And if you look on the data, you see that two inhibitors, chemostat and E64D, these inhibitors inhibited two different proteases. One is this TMPRSS2 already shown, and the other is the protease KTBL. And you see both alone, E64D not significantly, but chemostat yes, and synergistically block then this entry. And of course, there are more experiments in this paper, and they have also shown that, for example, acidification of this compartment is important to block entry. So this is just an example how basic research can identify potential points of attack. And well, the question is the former. And it's really a pleasure to say the former president of the United States considered, of course, himself also the #1 virologist. And you might remember what he suggested is use chloroquine. And is that completely mad that he should use chloroquine? Well, this is not completely mad, because chloroquine is used for malaria prophylaxis and treatment. But the one thing that chloroquine does, it inhibits endosomal pH acidification. And as I've shown you before, this is required for viral entry, and several viruses use this path. So in principle, there would be mechanisms which could be used against viral diseases, and it could be a potential broad-range antiviral activity. So the virologist #1 was not completely stupid. However, people who are specialists in the field and medical doctors, these two, for example, published in the Deutsche Ärzteblatt, a positive effect of chloroquine, this was in April, a positive effect of chloroquine for COVID-19, which was tested for decades in a multitude of viral diseases without success, would be the first of this kind. And indeed, it didn't work. So it didn't really work. Well, Trump got infected by corona himself. Was he treated with chloroquine? No. He was treated with remdesivir, and he was treated with an experimental antibody cocktail. Now, Trump, as you remember, was healed from corona, and therefore it looks like these therapies work. However, to be honest, you see that many people are still dying, also in developed countries where you have access to the full medical system. Of course, things have evolved, so I would say very simple therapeutic settings are standard now, where you block the inflammation in the lung. So the pathogenicity, one part of the pathogenicity is that if you have the virus in the lung, this leads to an inflammation. This leads to a thickening of the barrier between blood and the cell layer between blood and the air, where you have the oxygen transfer happening. And if this is thicker, the efficiency of the oxygen transfer goes down. So these people then need oxygen. So this is based on the inflammation. And if you block this inflammation, things can get better. So dexamethasone, a corticosteroid, is a standard treatment now in these cases. All other therapeutics, to be honest, including remdesivir, show some profit in some cases. However, it's not that these drugs are not like antibiotics for bacteria that you give them and everything is fine. So it means that the treatment approaches which are used and have been, let's say, tested for corona in the clinics, some patients profit, but the vast majority doesn't really profit from them. And this is, of course, a problem. So you see that the normal therapies don't work. And the thing which most likely worked with Trump was this antibody cocktail. This is quite expensive, and this was, of course, to that time experimental. And antibodies are quite promising because what you do is you block the viral entry, and there are a lot of companies working on that, including, for example, just as an example, Mimo Therapeutics, which is a company in Switzerland, and they have a special technology because they have a very high efficiency to extract the antibodies from B cells in humans. And I had a Prof's cast with the CEO of this company, Carsten Fischer, recently. This I will upload in the next days. And so what does this all mean? If I say antibody work, but not always, then we have to be cautious in the therapeutic setting, not always. However, the major part of the drugs at present are not really curing you. At the end, most of the patients get cured by the own immune system, and the intention of vaccines is simply to take the immune system to avoid the infection. So we have to have a look on how this works. Now let's look again how this viral replication works in a simplified term. So we have our coronavirus here, and now the first step always is this attachment, and then you of course this internalization and so on. But at the end, what happens is you have the release of the genetic material into the cell, and this genetic material then is of course, you have replication and so on going on, but at the end you produce more genetic material and you produce the viral proteins. And then you have the assembly, and then the virus gets out again. The details here are, of course, a bit more complex. We have seen that before. For developing a drug, the details are important. For developing a vaccines, the details of this replication cycles are not so important, because at the end now, if we ask ourselves, how can you now block or avoid a virus infection? Well, one thing is, avoid that the virus gets in contact with a receptor. And how can you do it? Yeah, neutralizing antibodies. So as soon as you have antibodies which can block this interaction with the receptor, the virus cannot get into the cell, and that's it. And viruses are not bacteria. Viruses need the host in order to replicate. If they don't get into the host cell, a virus is kind of dead. It can't do anything. Yeah. So therefore, just blocking for viruses is cool. However, if you have an infection, of course, ongoing, then the point is obviously that these antibodies are of no use. The virus is in the cell. So if you want to get rid of an ongoing infection, antibodies are not enough. You need also take care about the infected cells. And now what happens is in the cells, this is the spike protein, for example. These proteins are cleaved via the proteasome and then presented on the surface of the cells. You will, if you choose immunology as an elective molecule, you will learn that it's being associated with the MHC class 1 molecule. And this complex here, so this is the viral part and this is the presenting part, can present then these virus parts to cytotoxic T cells. And this is an interaction which happens via the T cell receptor. And this is specific. So these cytotoxic T cells then recognize a viral a cell infected by a virus and can kill the cell. So these are the two possibilities how our immune system can fight against viruses. We have this antibody part and we have this the CTL part. Now let's have a look, where do these cells come from? So at the end, you must induce these cells. And the problem is, at the beginning, you have to think of that each antibody or each cell which can produce an antibody recognizes something different. So we have millions of different, these are B cells, these are called B cells, different B cells in our body. And only one of 1 million, for example, recognizes the coronavirus. So if you have an infection and this cell is the only one and produces a couple of antibodies, this will be not enough. So the first thing what you need to do, as this as a low frequency, you need to activate this specific B cell, and you need to tell the B cell now proliferate. You need to be more before we can start doing things. And then we have high frequencies of the cell, and this process is called clonal expansions. Of course, also the details is very simplified. Details refer to the immunology lecture. And then we have a differentiation of these B cells. They produce then antibodies, they secrete the antibodies, and then we have this blockage. So this is what happens in the case of the antibodies. Now, what's about the cytotoxic T cells? The same problem. We have just maybe 1 in 1 million cytotoxic T cells, which are specific for the coronavirus. The others recognize different other things from other viruses, for example. So also here we need an activation and clonal expansion. However, for this to happen, the virus must first be in the cell in order to activate the cytotoxic T cell. Virus is in the cell anyhow, so this is not a problem. And then also here we have the high frequencies, and also then we have a differentiation into effector cells, which can actually kill the target cell. Now, this looks like, where's the problem? Well, the problem is this takes 14 days. And if you have bad luck and you have a severe disease, you simply don't survive these 14 days because these are cells. Cells have doubling times of maybe 1 day. So from here to here, this takes long until you get a high enough number of cells, and you have to survive this period. This is a problem, and not all of us do that. If you're recovered at the end, you recover because your immune system. And now what you can do is, of course, you can simulate the stimulation step at the beginning with a vaccination. So what do you need to do in the case of this? Well, you need just to have a protein, which is, for example, the surface protein of your virus, which then activates your B cells and which leads to this high frequency. In the case of cytotoxic T cells, of course, you need then this protein being inside of the cell. So just injecting the protein is not enough. And then you get your high frequency. And then you have your effector function, and you generate memory. So this can take a bit longer here. You generate then just memory, having not enough antibodies, for example, or enough active T cells around. And then this happens very fast. So this is how this protection happens. As you know, there are now already vaccines on the market, and some of them are in development. And now we want to have a look on how do these vaccines actually work. In order to understand what are the potential dangers and so on, you need to know how these vaccines work. So you need, in principle, we talk about RNA vaccines, and we have the AstraZeneca vaccine, we will talk in a second, and we have the typical vaccine class are just protein subunit vaccines. So a protein subunit vaccine, which is also developed, I think this is GSK Sanofi. You would just use the S protein. As we said before, what you want to have is neutralizing antibodies. So just use the S protein, formulate it appropriately, inject it, and then you will, of course, induce your B cells and you will produce antibodies. So this is a very simple, straightforward approach. Clone it, express it, and that's it. Now, an RNA vaccine is acting differently. An RNA vaccine consists of RNA encoding for also this S protein. The RNA is being injected, and then within the cell, the RNA is translated into this S protein. Whether it's the complete S protein or not, this is something else. And also the details, whether it's presented on the surface also, we don't care here. At the end, we produce the viral S protein here. Now, of course, you can secrete that or display it on the surface, and it will be recognized by B cells, and the B cells then can, again, like here, proliferate and produce antibodies. Of course, this is clear. So you can achieve with an RNA vaccine. But with an RNA vaccine, you can do more because the RNA vaccine is produced inside of the cell, and therefore this also can be presented to cytotoxic T cells and activate a cytotoxic T cell response. A DNA vaccine is similar. DNA vaccines are also directly injected into cells, and then it's first transcribed into RNA, translated into the protein, and the remaining part is the same. I worked myself with DNA vaccines for years. These were discovered before RNA vaccines. And DNA vaccines, you really injected naked DNA. Now, maybe one more word on and bit more detail on this RNA vaccine. Again, DNA vaccines, you inject naked DNA, and you inject per mouse, for example, 50 micrograms of DNA, which is a lot. Not per mouse, it's 100 per mouse leg. It's 50 micrograms. Now, the RNA vaccines, the dose of the Biontech vaccine is 12 micrograms, which is way lower. And RNA is way more unstable than DNA. So this will not work if you don't have a appropriate formulation. So let's have a look on the real vaccines. First of all, if you want to induce an immune response, you want to get to a special cell type, and this is called the dendritic cell. So you don't care about most of the other cells for your vaccine. You want to get to this dendritic cell because this is the cell type which can induce other cells. For details, again, you refer to the immunology lecture. Now, there you want to get. So therefore, ideally, you will have a formulation which makes this optimal to get into this type of the cell. And second, a formulation which protects your RNA from degradation. And therefore, this Biontech vaccine, which is an RNA vaccine, is in a lipid formulation. So it's not a virus particle. It's a particle. It's a lipid particle. This is optimized to avoid degradation of the RNA. If you just have free RNA, it will be quickly degraded, and to optimize this uptake by the dendritic cell. Now, the other vaccine, which has been approved, at least in the US and in the UK, is the vaccine of AstraZeneca. And this is an adenovirus particle. So this instead of a lipid, which is just a chemical lipid in this case, this is then a particle which consists of a virus. Adenoviruses are the cause of the common cold, so they're very benign viruses. You just have twice or 3 times a year you have an adenovirus infection and get your normal common cold, which is then after 5-6 days it disappears. It's not something which kills you. It doesn't make you happy, but it's not really a severe disease. And the adenovirus particle, which is used for this AstraZeneca vaccine, comes from chimpanzees. So it's not a common cold of humans, it's of chimpanzees. And second, this is manipulated in a way that it cannot replicate anymore. So you don't produce more, it cannot reproduce anymore. So you don't produce a more viral particle. It's just the particle which is used as a transport for your, in this case, recombinant DNA. It's not just it's a DNA virus. We have seen that at the beginning. It's also transporting the DNA encoding for the surface protein. So this DNA then gets into the cell, is transcribed into the RNA and produces the surface protein. The difference is only that you have a viral particle around carrying that. So, and now if we asked ourselves about efficacy, so this is the Biontech vaccine, the AstraZeneca vaccine, and we could say, well, a protein vaccine doesn't work so good because it only produces antibody and the other produces also the cells. So the question is, what do you actually need? And this question was already asked in the chat and already almost answered in the chat, and this is correct. So if we go back and have a look at the mode of action, we see that we have this part neutralizing antibodies and we have this part cytotoxic T cells. And indeed, if you're not yet infected, you do not have viruses within your cells. So if you achieve with your vaccine that no cells, no virus particles enter the cell, antibodies are enough. And this is indeed the mode of action of current vaccines, is just based on antibodies. The protein vaccines which are used do not induce any relevant levels of cytotoxic T cell responses at all. It's just antibodies. That's the mode of action of current vaccines. This works for most viral diseases. It's good that in principle this also works for corona, so you can go for a development in this direction. It doesn't work for viruses which infect immune cells like HIV or HCV and so on. And in this case, we have no vaccine yet. So if coronavirus would have a similar replication cycle to HIV and would be transmitted via the respiratory system, this would be a nightmare because vaccine development against such a virus would be way more demanding. So in this case, we were lucky. We have a virus which from the replication itself, it's clear that blocking the entry should be enough as for other viruses. And we are also lucky that this virus has for an RNA virus, quite low mutation frequency. quite low. It's not zero, it's quite low. The more people are infected, the higher the likelihood that you have mutations, and the likelihood increases that you have mutations which might, where your vaccine might not be protective anymore. So this risk exists, and therefore you need to keep this frequency low as long as you have not vaccinated vast parts of the population. With respect to efficacy, another thing is, so of course, if a vaccine needs to be effective, I need to have enough antibodies. So I need to block all of this surface proteins and not just parts of them, because if it's only parts of them, it's just no more protective, full stop. So I need to put all of them around. Second, the point of viral entry is the respiratory system. So I need the antibodies to be there. I need to have antibodies which are in the mucosa. This is also something, this is a special isotype, IgE antibodies, for example, which are also not so easy to measure. Predicting what if you come up with a new vaccine, and there are not so many vaccines against cold viruses. You have an influenza vaccine, but others are not there. So predicting how good an approach will work is not so easy in this case. So therefore, it's a very wise idea to develop many vaccines in parallel, which are based on different mode of actions, and then look in the clinical trial at the end how they work. It's very important that you have that you rely then on the data on the clinical trials. So how does an effective vaccine look like? So we have here an example of a vaccine which is perfect. So we have, we need, as we discussed, we need a certain antibody level in order to protect. And if this antibody level is high enough, people are protected. As soon as it's lower than this threshold level, you're no more protected. So ideal vaccine, you vaccinate once and then you have a lifelong immunity. This you often see with live vaccines, which are vaccines based off viruses or bacteria, which are just attenuated but still can replicate. The typical profile of subunit vaccines or killed vaccines, this will also most likely be the profile of any vaccines which cannot replicate, like RNA vaccines, or also the AstraZeneca vaccine, which looks like a virus, but it's just the particle it cannot replicate anymore in the host cells. So most likely these vaccines rather will have a profile like this, that you vaccinate, that the antibody level will decrease, and it will decrease after some years to a level below the threshold level. So in order to keep your protective effect, you need to vaccinate again. This is what you shouldn't forget also for other diseases like measles and so on. You should check whether you have to repeat your vaccination. So, and then you're again protected, for example, for 5 more years, and then you would have to do it again. And if you forget it, you will not be protected anymore. Now, the initial vaccination schedule is usually such a schedule like this, that you give a prime vaccination and then you boost after, for example, 3 weeks. And this prime vaccination itself only leads to low levels of antibodies, and then you boost and then you can come up with very high level. Of course, so 2 to 3 weeks after vaccination, you have already certain levels of antibodies, and some of the people might be protected, and there are not studies looking at that. And it looks like 50-60% of the people with the Biontec vaccines are already protected after the first dose. However, this is small, and it's likely that this is not so long lasting. So therefore, this boost is, of course, required. And then you're here. So this is how these vaccines work in principle. So the vaccine efficacy, of course, is the thing you want to know. You can predict a lot or so, but at the end, what matters are the clinical data. So you need to vaccinate the people, and you need to have a look on that. And for example, the Biontech vaccine, 21,720 people have been vaccinated in the phase 3 study with the RNA encoding for the viral surface protein. And there were 20,000 people, another 20,000 people, which were vaccinated with a control. So this is 20,000 people have received the vaccine, and the efficacy for the Biontech vaccine was 95%. And this is, to be honest, this is amazingly good. Also, I was surprised, and I was really, when I have seen this result, I was really, really, really happy. And again, congrats to the team of Biontech. This is really incredible, very good work. They took the risk and so on. So this is really, really, really very good. And this efficacy, of course, was surprisingly high, but of course, this is cool because this was the first vaccine which was developed, and you could start right away. Unfortunately, the BioNTech vaccine has to be cooled down to minus 70 degrees. So this is a vaccine which is not really suited to be distributed in developing countries where you don't have the infrastructure. This would be extremely difficult. So the AstraZeneca vaccine is way simpler, cheaper, and can be stored simply at 4 degrees, which makes all the logistics way easier. BioNTech vaccine to be celebrated. What about the AstraZeneca vaccine? It's 70%. 70% sounds not good compared to 95%, but honestly, this is also good. So the 50% was set as the let's say limit of the WHO for vaccine efficacy, and it's 70%. So that's well beyond. If this would be the only vaccine, everybody would be also completely happy and would have celebrated. So everything is fine. The interesting thing about the AstraZeneca vaccine, and you see that there is some space here, is that in the case where the first dose was injected, the complete first dose was injected. So they are also scheduled with 3 weeks into, I think it's also a 3 weeks interval. And usually you would inject the first and the second dose the same amount, so 100% first dose. And then the efficacy was lower. This was the Brazil part of the study. And occasionally, unintentionally in Britain, a lower first dose was injected. So only 50% of the intended dose. And interestingly, this was more protective. So for a conventional drug, this would be completely unintuitive, because a conventional drug, usually you have a dose response which follows the dose. So the higher the dose, the better the protective effect. However, also the higher the side effects. Now, this is the other way around. And the question is, what happened here? So it's good, first of all, it's good that this unintentional dosing happened because you can learn something of that. It's not something you would trust in the clinical trial team. You would say this is not something which is really ideal. It should not happen, to be honest, but it happened. And then as it happens, it's wise to have a look on the data, and most likely what the reason is. So this is the curve you have already seen, and this is most likely also the 50% priming curve. So you induce a small response at the beginning and then you boost it. Now, what most likely happened with a 100% priming schedule is that, of course, you produce a bit more at the beginning, but with a boost, this went rather down than up, just in the extreme. So it suddenly went up, but not to the same extent, but just make it here to make it clear here. So it's lower than what you, at least what you would have with a lower dose. And the question is, why does this happen? Well, remember, the AstraZeneca vaccine is not a naked RNA vaccine. In the RNA vaccine, you just have the RNA which produces the protein which can induce the immune response. Now here you also have a carrier. And of course, you will also get a response against the carrier. And if you have a priming which with a high dose, you will have more antibodies also against the carrier. And you see that the carrier, at least to my knowledge of the AstraZeneca vaccine, doesn't express the surface protein. So you could design a viral particle also that the surface protein of the coronavirus is expressed on the capsid, which is here not the case, which is, by the way, also not so easy for a non-enveloped virus, because this is completely unrelated. So the problem is that if you have then, after the priming, antibodies against the carriers around, you can capture these carriers by the B cells, and you will boost your response against the surface protein of the carriers, but not against the S protein. And therefore you have then this discrepancy here. And therefore this is a very important lesson and very interesting that we know that now, because obviously you would go now for this 50% dose. That makes sense. And now comes the point, why is this not yet approved in the European Union? Are they so stupid? Well, the problem is that the number of people who have received the first dose is here just 1,370 of this 50% dose. So combined, everything is fine, but just this group is quite low. If you compare that here, you have 21,000 people. Here it's just 1,300 people. So the statistics here is weak. So therefore, agents like the EMA say, before we can approve that, show us more data for this patient, for this group with this dose, and then we can approve it. And this is the proper way how to approve a drug, because you don't want to have an approval based on an effect which is at the end statistically not relevant and it's lower, and then you bear a risk that, for example, the protection in other age groups also is lower. So we talked about efficacy, but the other thing is safety. So in other words, both vaccines at level on the data are effective, and both vaccines with respect to efficacy are highly recommendable, full stop. This, by the way, I always talk from the Biontech vaccine. For the Moderna vaccine, it's the same. It's also an RNA vaccine. The efficacy is comparable to the Biontech vaccine. So it's no matter all these three vaccines, which are now approved. AstraZeneca is not yet approved in the EU, but in the US and UK. I would, based on the efficacy, take it immediately. What about safety? So for sure, you could start in May, when I first talked about that. There was no data, so therefore you could only speculate. But now we have the data, so we don't need to speculate based on the mode of action. From clinical trials, there is simply no unexpected side effect. And remember, in the clinical trials with the Biontech vaccines, 20,000 people have been vaccinated, and this is what is exactly what you would expect from a normal clinical trial. You would have vaccination of people around that. So it's not less than you would have in a regular vaccine approval process, because some people always think not enough people were included. No, this is not true. This is exactly the amount of people you would have in a normal approval process. However, you have packed these people, you have packed the complete development path, and therefore it made it faster, and you have taken the full risk. So these Biontech guys have taken the full risk for the development. If the vaccine would have proven to be ineffective, this might have killed the company. So this is what I have to point it out. So it's not that you can lean back and say, that's it, but you take the risk. And only then this development is so fast. But you could say, well, of course, you couldn't observe the people for long term after they have been vaccinated. And this is, of course, a difference to a normal trial where you would wait longer. However, here you see the efficacy, you don't see a safety signal. So it's wise to start right away with a vaccination. Because even if there would be a long-term signal, if you outweigh that with the problems of your pandemic, this is not a real risk. And of course, you have experience with other vaccines. So what about the RNA itself? So it's an RNA. So therefore, this genetic, some people are afraid that you have a genetic variation and so on. It's RNA. It doesn't integrate into the genome. It has to be, if you are an RNA virus and you want to integrate, you need to be a retrovirus like HIV, and then you have a reverse transcriptase, and then you might be able to integrate. You have also integration sites and so on. But it is not a retrovirus. So this is RNA, and RNA does not integrate into DNA. Full stop. This is science. It's not a question of it could or no. No, it doesn't. Full stop. The virus, the DNA virus from AstraZeneca, it's derived from a common cold virus. We have 3 times a year common colds, but people do not die due to lung cancer due to common colds. Never. They die due to lung cancer due to smoking. So also this virus from the particle itself, there is no, based on the mode of action and of the structure, it's not foreseeable that you have a genetic modification or cancer whatsoever. Autoimmunity and so on is always something which could happen, and there are rare cases of autoimmune reaction, Guillain-Barré syndrome, and so on, of also from vaccines. I expect that it's not proven, and of course, these cases, then the frequencies are so low, then given a causal relationship because between very rare autoimmune disorders and vaccination is difficult. Now, another thing is allergic reactions. So this is also something what you would expect in the case of vaccination. You stimulate your immune system, and if you have someone who is highly allergenic, the immune system is already biased. So what about this allergic reaction? Now, in the clinical trial, what you do is you usually exclude people with a history of severe allergic reactions because you don't want to have that in a trial because you are already expect that you might have some problems there. You don't want to have that in the study where you don't know whether there are also other side effects. So there was in the trials, there were no unexpected frequencies of allergic reactions. Later on, it turned out that it seems that the frequency of allergic side reactions with the RNA vaccine might be a bit higher. Again, might. It's not that this is something which is, to the best of my knowledge, already statistically significant. However, it's still in the window of the expected reactions. Let's say the reaction in this case, an immediate reaction, has to be then against the lipid part of the vaccine because RNA itself is not immunogenic. So, of course, this is being looked at. You would say, and this is what Norway, for example, does, if you are now 95 years old, almost moribund, then a vaccination might not be a good idea. It's not that you have allergic reactions in this case, but then you might not survive the very mild fever also you induce normally with a vaccination. And these people then you have to weigh it whether the risk of being infected with a coronavirus, and which certainly will kill you, by the way, if you are in such a condition, is tremendously higher than the risk of you, this special subgroup, of being killed. But if you are healthy, then you will not be killed. You will not be killed by this little fever and side effects. We talk about more or less moribund old people. Yeah. And the allergic side reactions, what were observed, the vast majority are observed in patients and in people with a history. So of course, the vaccinations are done, and this usually happens immediately. So, and you can handle that if you are then in the hospital and they have their adrenaline and so on. So this is something which does not necessarily kill you, but you shouldn't vaccinate yourself at home without a supervision, especially if you have a history of severe allergic reactions. Okay, now, so at the end, this is what I learned from our last discussion. So this is a picture of parts of my family, at least my kids and my mother and my mother-in-law. And my mother has been vaccinated January 7th, and my mother-in-law has been vaccinated January 12th against coronavirus, using the normal process in Baden-Württemberg, registration and so on. So not something which is exceptional. My mother-in-law was told by her doctor she was the first one of her village being vaccinated. So my kids, of course, are not vaccinated because they're, as you and me, too young. So this means too young, not for to getting the vaccine, but they're not yet. But they are vaccinated against all disease. The STIKO recommends, of course, as myself. I want to take this for some thoughts. If you look at this criticism or skepticism with respect to vaccination, it's amazing. I studied at an elite Grande École in France. I worked for many years in vaccine development. I made my PhD on the most renowned Institute of Infection Biology in Germany, the Max Planck Institute for Infection Biology. I worked, my diploma thesis I did at the #1 vaccine developer for childhood vaccines in Siena, Karen Biocchini. Many of the childhood vaccines were developed there. I worked in the pharmaceutical industry working on vaccines, in this case, therapeutic vaccines against cancer. And I readily looked at my mother, and my mother-in-law gets vaccinated against corona. As soon as it's my turn with respect to the waiting list, I will as soon as possible get the vaccine. And the same is true, of course, for my kids. And it's amazing. I'm really trained for years in this field, and if I consider myself as an expert, then I consider myself an expert in the field of vaccinology. I was head of vaccine development and so on, and I know how things work in the pharmaceutical industry. But it's bizarre. People now who watch for 2 hours of YouTube video, now for them it's clear everything is wrong. And me studying for years in this, I'm too stupid to recognize that everything is wrong. That's amazing. But this is the problem today, that you cannot talk to these people because they don't listen to you. They don't want to listen about the details. They don't want to hear about in the lung you need IgA antibodies or whatsoever. But they tell you, no, I'm sure all this vaccination stuff is wrong, or the coronavirus doesn't exist even. So this is really bizarre. And you, as my students, you become scientists. You have to base things on science, and you can draw, of course, different conclusions. So for an example, we had a discussion in the 3rd semester current topics of biology with my friend and colleague, Professor Florian Wichern. And he is a soil specialist and, of course, connected to agriculture. And with respect to transgenic plants, especially here in Germany, everybody agrees now in science that they don't harm you, and also for the environment, it's not a problem. So therefore, condemning transgenic plants per se is bizarre. However, of course, you could discuss about risks with respect to monopolies and so on, and draw different conclusions. That's okay, and you need to discuss with these people. But all of this debate is based on scientific facts. And you simply draw different conclusions. The same for nuclear power, for example. Also, these discussions were based on scientific facts, but the conclusions drawn were different. In the case of the vaccine skepticisms, this is not based at all on any scientific facts. It's just bullshit. Full stop. And of course, you could discuss about what is the likelihood of autoimmune, risk of autoimmunity and so on. As long as the data is not there, I cannot say that, but based on experience, it would be very, very surprising that such a vaccine would have that you would provoke millions of people with Guillain-Barré syndrome, because there are other vaccines already on the market where you don't see this. Yeah? And therefore, this is bizarre. And of course, as I say, you cannot exclude it because simply the amount of people has not been vaccinated. But this is really bizarre. Yeah. So, and saying that the vaccine itself doesn't work, or so a vaccination itself cannot work. This is amazing. Let's see whether there are more questions. The question is, do drugs against viruses have something to do with the CTL cells? Do they accelerate their job? This is a very good question. In the case of viral infections, rarely, but there are drugs on the market for cancer treatment, which are so-called checkpoint inhibitors. And there, 2 years ago, Allison and his colleague got a Nobel Prize for discovering these. It's a CTLA4 and PD-1 inhibitor. And these checkpoint inhibitors indeed foster cytotoxic T-cell response. Usually, however, this is, to the best of my knowledge, they are not used in viral infections. It's just, again, in cancer. You also have the problem that the cell is not active enough. So in the case of In the case of the viral infection, it's more a race. So the problem is more that the let's say the building of your antibodies is not of your antibodies and cytotoxic T cells. So until you remember this clonal expansion phase, this takes too long, and this you cannot accelerate because it's just the proliferation of the cells. So therefore, in the most viral settings, acute viral settings, such an drug which interferes with T cell development or activity is not really a big help, I would say. Yeah, it depends on the virus. If it's especially interfering with the immune system, it takes longer, and then this could be different. But a typical viral infection, I would say, not really of use. And these drugs are, of course, dangerous because they can also they have really severe side effects because you can interrupt also the self-recognition, and then you will induce autoimmunity. And this is not rare in this case. So if you use these drugs, it's quite common that you have severe autoimmune side effects. And yeah, I hope now we could make it a bit clearer how coronavirus looks like, how the vaccine works, what about efficacy, what about safety. And I hope that your fears are gone getting a vaccine shot. It's the best you can do. And thanks a lot for your attention. Stay healthy and goodbye.